Introduction. Welcome to today's lecture on cellular respiration. In this session, we will explore the intricate processes that occur within cells to generate energy. Cellular respiration is a fundamental process that allows organisms to convert nutrients into usable energy. We will delve into the various steps and components involved in this highly regulated and efficient process. So, let's begin our journey into the fascinating world of cellular respiration. Glycolysis is the first step in glucose breakdown, occurring in the cell cytoplasm. It converts glucose into pyruvate, producing a small amount of ATP and NAD. Key steps include glucose phosphorylation by hexokinase, trapping glucose in the cell, and the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1, PFK1. PFK1 is regulated by ATP and other metabolites. The citric acid cycle, or Krebs cycle, occurs in the mitochondria and fully oxidizes acetyl-CoA to produce high-energy electron carriers. These carriers are important for cellular respiration. Acetyl-CoA in the cycle generates NAD, FOD2, ATP, and other intermediates for metabolism. The electron transport chain is located in the inner mitochondrial membrane and is responsible for the transfer of electrons from NAD and FOD2 produced during glycolysis and the citric acid cycle. This transfer of electrons occurs via a series of membrane-bound protein complexes, complexes I4, and mobile electron carriers, such as ubiquinone and cytochrome C. The final electron acceptor is oxygen, which, upon accepting electrons and protons, forms water. The electron transfer within the electron transport chain generates a proton gradient across the inner mitochondrial membrane. Oxidative phosphorylation is a process that utilizes the proton gradient across the inner mitochondrial membrane to generate ATP. In this step, ATP synthase, a complex enzyme, facilitates the flow of protons back into the mitochondrial matrix, coupled with the synthesis of ATP. This process is highly efficient, as one molecule of glucose can generate up to 30 to 32 ATP molecules through oxidative phosphorylation. The energy stored in the form of ATP is readily available to perform various cellular functions. ATP synthase is a motor-like enzyme complex that produces ATP by utilizing the proton gradient generated by the electron transport chain. It is composed of two main parts, F0, located in the mitochondrial membrane, and F1, located in the mitochondrial matrix. Protons passing through F0 induce a structural change in F1, allowing the conversion of ADP and inorganic phosphate into ATP. This process, called chemiosmotic coupling, exemplifies the connection between energy transduction and biochemical reactions. Cellular respiration is tightly regulated to meet the energy demands of the cell while avoiding excessive production of reactive oxygen species, ROS. Several key enzymes involved in glycolysis, the citric acid cycle, and oxidative phosphorylation are subject to both hormonal and allosteric regulation. For instance, phosphofructokinase 1, PFK1, a key regulatory enzyme in glycolysis, is regulated by ATP, ADP, and various other metabolites. The regulation of cellular respiration ensures that energy production is balanced and adjusted based on the cellular metabolic requirements. Mitochondria are double-membraned organelles that serve as the powerhouses of the cell. Their outer membrane allows the exchange of metabolites, while the highly folded inner membrane, known as cristae, houses the electron transport chain and ATP synthase. This unique structure maximizes the surface area for ATP synthesis. Mitochondria also possess their own circular DNA and ribosomes, as remnants of their bacterial origins. Mitochondrial dysfunction can lead to severe consequences, as observed in various mitochondrial diseases. During cellular respiration, a small proportion of electrons leak from the electron transport chain, leading to the generation of reactive oxygen species, ROS, such as superoxide anion, O2, and hydrogen peroxide, H2O2. While ROS play important roles in cellular signaling, excessive production can lead to oxidative stress and damage cellular components, including DNA, proteins, and lipids. To counteract this, cells have evolved various antioxidant systems, including enzymes like superoxide dismutase and catalase, to neutralize and scavenge ROS. Cellular respiration is intricately interconnected with other metabolic pathways within the cell. For example, the intermediates of glycolysis and the citric acid cycle can be utilized in the synthesis of other important molecules in the cell, such as nucleotides, amino acids, and lipids. The integration of different metabolic pathways allows cells to respond to changing metabolic demands and maintain homeostasis. Dysregulation of these pathways can lead to various metabolic disorders and disease states. Alterations in cellular respiration and metabolism often occur in diseases, such as cancer, diabetes, and neurodegenerative disorders. Cancer cells, for instance, exhibit altered metabolism, known as the Warburg effect, wherein they rely predominantly on glycolysis for energy production, even in the presence of oxygen. Such metabolic alterations provide survival advantages to cancer cells and influence tumor growth and progressions. Understanding these alterations in disease states holds promise for developing targeted therapeutic approaches. In the absence of oxygen, some organisms can carry out anaerobic respiration to generate energy. Anaerobic respiration utilizes alternative electron acceptors, such as nitrate or sulfate, instead of oxygen. Although anaerobic respiration is less efficient in terms of ATP production compared to aerobic respiration, it allows organisms to survive under conditions where oxygen is limited or unavailable. Various bacteria and archaea are capable of performing anaerobic respiration, showcasing the diversity of metabolic strategies in different organisms. During exercise, the energy demand of muscles increases significantly. To meet this demand, several metabolic adaptations occur within the muscle cells. These include increased glucose uptake, enhanced glycolysis, and mobilization of stored energy sources, such as glycogen and fatty acids. Additionally, there is an activation of specific transcription factors that regulate the expression of genes involved in energy metabolism. Understanding these adaptations can have implications for improving athletic performance and designing exercise interventions for various populations. Process that allows organisms to produce ATP. Process involves the breakdown of glucose, the citric and oxidative phosphorylation. Understanding pharmacology and the changes associated with disease and exercise.